My name is Duval, and I'm a professor here at Colorado State University, and I'm the director of the Molecular Oncology Lab in the Flint Animal Cancer Center. When I was an undergraduate, I was a biochemistry undergraduate, and I worked in a research lab that looked at natural products and how they could alter cellular metabolism as a way to treat cancer. So I started off working in cancer research. From there I kind of moved on to looking more at gene regulation. And so now working here in this lab where I can kind of put those two things together, it's really just an ideal environment for me. Our work here is really trying to identify how human cancers and dog cancers look alike at really the most basic fundamental level, right down at the very genetic changes that occur in dog cancers. Right now we're looking at 4 major cancers here in the lab. We're looking at bone cancer or osteosarcoma, we're looking at bladder cancer, soft tissue sarcomas, and then also thyroid cancers. And what we're trying to do is identify the genetic mutations that drive those different cancers. We kind of live in this remarkable time where, following the Human Genome Project, the ability to sequence our entire genome has just exploded, and it's become affordable. We've been working with CRISPR-Cas9 systems, and these are gene editing systems. So we can basically go in and knock out one gene at a time across the entire genome. And we can ask what happens when we do that to those cells. This allows us to target individually, 30,000 different genes and ask how they contribute to the development of the tumor, or maybe how that tumor can spread to a new site. This is important because until recently we had hints, but we really didn't know exactly what were those fundamental changes in the canine cancers. So we're going through and we're using this huge next generation sequencing to identify those little tiny changes in genes that allow the cancer to develop. And then we can compare those to how human cancers develop and put that information together and decide, how best to treat both human and dog cancers that share those same types of genetic changes.